Hi, my name is Debbie Stevens, and I'm the director of the Chronic Lymphocytic Leukemia program at the University of North Carolina. I'm very pleased today to be joined by Dr. Nitin Jain from the MD Anderson. So if you could introduce yourself and tell us your role there.
Sure. Excited to be here with all of you. Myself, Nitin Jain, I'm a professor in the Department of Leukemia here at MD Anderson. I treat patients with CLL, also acute lymphoblastic leukemia, and also I'm the director of our CAR-T program for the leukemia group here at MD Anderson.
Great. Well, thanks so much for being here today. We'd like to highlight some research that you and your group have done on a phase 1 study. And this is a phase 1 study of SC291, which is an allogeneic anti-CD19 CAR T cell. The design of the study is meant to be for all patients with relapsed or refractory B-cell malignancies. So I want to start out just simple, if you could just explain to me what is CAR T cell therapy?
Sure, yeah. So, CAR T cell therapy, I think, is a big revolution in the field of leukemias and lymphomas. And I think in CLL, it has a bit lag behind, but, we'll talk about maybe what are the issues there. But basically, the concept here is that you take a T cell, which is a normal immune cell, either from a patient, which is called the autologous CAR T cell therapy, in which several versions of which are FDA approved, or you take from a third party, like another human being or a cord blood or some kind of a stem cell derived T cell from another person, and that's called allogenic. And basically, you take these T cells, you take them in the lab, spend a couple of weeks, engineer them into supercharged T cells, which have done a gene called CD19 or something similar inserted into them, and then they will infuse back to the patient. And now these T cells are basically more laser focused. So rather than now they are just going to focus on killing any CD19 positive T cells or CD19 positive cells, and CLL cells and other lymphomas and many of the leukemias have CD19 on their surface. So that's basically the genesis of CAR T cell strategies. It was first approved in 2017, I believe, for children with leukemia, also with adults with lymphomas. And now we have seen many other disease indications, including in myeloma. I think CLL is maybe coming along now, finally, but I think it has been now in the clinical arena since 2017.
That's great. And I love this approach to therapy because it's really using the patient's own immune system to try to fight back against the cancer. And as you just mentioned, CAR T cell in the United States has been approved for several different indications, another type of leukemia, acute lymphoblastic leukemia, several different subtypes of non-Hodgkin lymphoma, but still is being considered for the approval in CLL. And so I guess from my standpoint, CAR T has really been variably successful in patients with CLL. And I was just wondering if you can describe some of the reasons it hasn't worked as well for all patients with CLL.
Sure, yeah. I think if you go back, maybe a decade ago, one of the first trials actually in the context of leukemias, lymphomas, done from University of Pennsylvania actually was in CLL. And, back, I think, 10, 12 years ago. And there was a lot of hope at that time that, CLL could be the first disease where this CAR T will be developed. But then I think from another thing which was happening in CLL in parallel around the same time was development of BTK and BCL2 inhibitors. So I think a lot of focus switched to that oral targeted therapy, which obviously works very well. And I think a lot of focus of investigators, the pharmaceutical sponsors and companies focused on acute leukemias as well as in lymphomas. Also, I would say at the same time, I think, some of the data we were seeing from CLL patients was maybe not as, the efficacy data was maybe not as good as was being noticed in lymphoma and acute leukemia. I don't think we clearly know why that is, but one of the potential reasons could be that, at the end of the day, you take patients' own T cells for most of the CAR Ts and you make them into a CAR T cell. And we know with research over the course of last couple of decades that the T cells in patients with CLL are dysfunctional. They have high expression of checkpoint markers and other immune dysfunction. And in the past, we and many other groups have looked at strategies to improve this T cell function by using checkpoint inhibitors or lenalidomide. But that concept remains that the T cells for patients with CLL are dysfunctional. So if your raw material you're going to make your CARs out of is not a so healthy T cell, I think that is bound to have some effect on the efficacy, and that's what I think we have seen in some of the published data with CAR T cell. And I think that also brings us to this allogeneic platform where if you were to derive CAR T cells from a healthy donor, presumably a person who is healthy, their T cells are healthy, they have no disease, and you use that to make CAR T cells, the raw material, possibly that would lead to a better efficacy of these cells. So I think that's where the role for allogeneic CAR T come about, that in the context of CLL, but it also makes things, if it were to be successful, much easier from a logistics standpoint, because then these cells are already pre-made from healthy donors. You don't need to collect the patient's own T cells. You can just, whenever a patient is ready to get CAR T cell therapy, you have it in your freezer, stored in your lab, and you can give T cells to the patient. So there are a lot of advantages of allogeneic T cells, and it's a field which is emerging now with several ongoing trials.
Yeah, and I think that's a great segue into coming back to talk about this SC291 compound that you have been working with. And so I guess in the simplest terms, I mean, you already mentioned it's allogeneic, so it's something that is made from healthy donors. Are there any other specifics about how this particular CAR T product works?
Yeah, so I think one of the challenges, this allogeneic CAR T cell field has been, in clinical trials with other products for several years now, since I think 2015, 2016. And some of the early results with many of these different products, not in CLL, actually, most were studying acute leukemia, ALL, and also in lymphomas, were not as good as we were seeing at the same time data from patients' own T cells. So now one of the issues when you infuse CAR T cells, which are derived from another person, is that these cells don't last very long. And generally your own immune system, patient's own immune system, will recognize these cells as foreign, which they should, and because this is a foreign cell, and they will reject these cells within a matter of 2, 3, 4 weeks. So they don't last for months on, they just last maybe for 3 weeks or 4 weeks. And so a lot of effort has been done in the last some time, how we can increase this persistence, at least maybe by 1 or 2 months, or maybe longer. And different companies are using different approaches. And this SC291, which is by a company called Senna Therapeutics, the lead investigator for the trial is actually Dr. Satwa Nilapu, who is at our center, vice chair of our lymphoma group. But the concept here is that they have done further genetic manipulations on these allogeneic CAR T cells to make them resistant or less likely to be killed by the patient's own T cells, NK cells, macrophages. So hope is that these cells will last longer and thereby will provide better efficacy than what we have seen with the previous version of these allogeneic T cells. So that's what, it's a trial. It's an ongoing trial right now, open at several centers in the United States, including patients not just with CLL, but other like mantle cell lymphoma, follicular lymphoma, and other B cell malignancies.
Yeah, this is great. And so it's kind of taking the parts maybe that worked with the currently approved commercial products, like it's still a target, it still targets CD19, but it really just tries to use a healthy immune system, healthy T cells to do the same thing that you're trying to do with commercial CAR Ts. So I know excitingly, the first patient on this clinical trial was a patient who has CLL and is actually one of your patients. Are you able to share how the patient did on this study?
Sure. I think at the ASH meeting, we updated the data for this particular patient. So this patient did achieve what is called a partial remission, which is, more than 50% improvement in adenopathy and the blood counts by day 30. And subsequently, this patient, we are trying to see what's going on in long term in terms of follow-up. There have been other patients which have been enrolled, both with CLL as well as with other disease histologies, including mantle cell and follicular lymphoma. But these patients are still being monitored early on the disease follow-up right now. And I think we're expecting, either later this year to submit an update of the clinical data with all these patients to a medical meeting, to to. But again, right now it's an ongoing phase 1 study. And, so we are kind of anxiously looking to see how the disease and the results will pan out for the patients.
Yeah, no, that's great. So it sounds like you're seeing some early evidence maybe that this works, especially for folks with CLL, but the number of patients is still small. I know in your ASH presentation, it mentioned that the investigators took some immune cells from the patient in order to study the interactions between the CAR T and the patient's immune system. Did you learn anything specific from these experiments?
So I would say, these are ongoing work right now. So there is a lot of effort being done through the pharmaceuticals, the sponsor, the Summit Therapeutics, to collect the blood and the bone marrow as well as the lymph nodes, if possible, of the patients. Try to see, first of all, the expansion of these CAR Ts and how these CAR Ts are interacting with the patient's immune cells. As I mentioned, this CAR T is designed to evade the patient's immune system so that they can last longer. But what that's really happening in the patient, right? So I think what are the interaction with NK cells, the macrophages, the patient's own T cells. So those are all being studied right now, I guess, are analyzed right now. At ASH, there was some brief data or very data was presented, but we only had a handful of patients enrolled at that time and still in early follow-up. But we're hoping now that, towards the middle or the end of this year, we'll have much more data to share.
That's great. So I guess the summary of this, exciting CAR T product is that this is an allogeneic product. So one that healthy donors help patients with CLL to try to use that part of their immune system to fight the CLL. Early data showing some evidence of response, and there's going to be more data to come. And so this clinical trial is called the ARDENT study. And you've mentioned it's an ongoing study. So how could patients find out if they were a candidate for this trial or more information?
So, this trial is open at several sites in the United States. I think one would be if you were to Google ARDENT and clinical, or go to clinicaltrials.gov, which is a website where all the clinical trials are listed, you'll be able to see the trial, the eligibility criteria, but more importantly, the sites in the country, in the US, where this trial is open. So I think the first thing will be to reach out to one of those investigators. Obviously, you can email me directly as well, and I'm happy to help coordinate that to see whether, depending on your location, whether there is a site close to your center or your place of living, or versus you want to come down to Houston. But certainly, I think right now it's open in 7 or 8 different sites in the United States. And as I said, it's also for CLL and other disease histologies.
Okay. And this, again, this is for patients that their disease has relapsed after prior therapy. So it's not for frontline treatment of CLL, but so reach out either to your oncologist or, look on clinicaltrials.gov if you're interested in this study. But I just wanted to thank you again for your presentation of research and your ongoing work in the field. It's great to have you speaking to all of us today.
This was wonderful. Thank you.